I'm doing, produce all of this data from people as you, students. And well, let's see what happened. Well, I can start telling you that in my lab is interesting to understand what happens in our brains physically and chemically when we build memories, when we form, when we encode information that we will use in the future, and will be essential for our lives. And we, as you know, we focus at the molecular levels mechanisms of this, how we form memories, and we choose the epigenetic mechanism, the epigenetic and synaptic mechanisms. We know that these are very complex systems, but we think there is some specific rules in this system that will help us to predict how they work. And okay, so the outline of the talk will be such as this. I will first tell you why it's important to study molecular mechanisms of memory besides the scientific curiosity, why it's important for the society nowadays to study these mechanisms. Next, I will tell you what are the functional units of memory. I will talk about circuits, synaptic circuits, neuronal circuits. And finally, in the last part, I will talk to you about recent data from my lab, in which we are studying the epigenetic control of memory. We focus on one specific epigenetic mark that it is, we think it's controlling memory. And finally, I will show you how and why it's important to develop novel methods to study. We develop a method that it is called, we name it nucleosynapse tag, that allow us to study epigenetic modulation of neuron activity and synaptic remodeling in the same time, in parallel. So it is something that can be done, was possible in the past. And okay, so okay, why it is important to study memory in these days? Well, as you know, there are a lot of people with suffering from dementia. Dementia is an umbrella concept. There are different types of dementia. The most frequent dementia is Alzheimer's disease. It's about 70% of people with dementia has Alzheimer's disease. How is defined dementia? Dementia is defined as a progressive or persistent loss of intellectual functioning, especially with impairment of memory and abstract thinking. So there are a lot of symptoms and signs, but these are the most frequent. And as you know, there is a dramatic increase in the number of people suffering from dementia. 10 years ago, we have 50 million. This M means, this M means millions. So we have 50 million people suffering from dementia, and this is really incapacitating for the everyday, for the living, all the activities we do every day. Okay, so the main domain in the cognitive domain affected is memory. So it is why it is not surprising to see this is what I'm showing here is a brain from a normal people. You can see how is the volume of the different two very important areas for brain functioning, and how it looks a brain from with Alzheimer's disease. You can immediately detect that it is a loss in the cortical region. And cortex, it's important for language processing, for abstract thinking, for the really high-level functions that we have as humans. And also, there is an almost loss in a region known as hippocampus. Hippocampus is critical for memory. If you remove the hippocampus from an animal, even from a human, it cannot, this animal or this human, it's unable to form new memories. So he will remember all its life, its past lives, but it is for him it's impossible to form new memories. So if you are interested in know what happens if we lose the hippocampus and we cannot build or construct new memories, you can see this movie. It's a really for sure you know Christopher Nolan is a famous director, and his first movie was Memento, and it's about a person that cannot lost his hippocampus and cannot build new memories. So he has to find ways to remember things like taking a lot of pictures, notes, making tattoos, everything to try to remember things that happened recently in his life. So how scientists think based on evidence memory is codified in the brain. So we know that memory is encoded in neuronal circuits, but basically there is a key concept that it is, we think that a key component, a functional unit for memory at the level of circuits is neuronal ensembles. Neuronal ensembles are groups of neurons displaying recurrent patterns of coordinated activity, and they, as I tell you, are the functional units for memory. How they work? So just imagine that these are the neurons in one brain. For sure, we have more than this. But if we were the total number of neurons in our brains, they can be in a low activity state or in a high activity state. So when we encode some specific information, some phase, some memory, some cell number, anything we want to encode in our brains and for a memory, the activity of a group of neurons increases. And it is higher compared with the rest of the neurons. So here is where the information is encoding in this group of neurons. Over time, in a process known as a consolidation, the activity of this group of neurons decrease, but never reach against the basal state. So they get activated synchronously each time we retrieve all that information. Each time we remember that information, the activity of this group of neurons increases again. Okay, and thereafter there is a process known as reconsolidation, or more currently known as updating. When we have a memory, scientists, we think that to add new information, we update this circuitry, and for example, we can add another neuron to the circuitry that instead of remember only the color of a car, we can remember the model of a car, or we add additional information. So, but the basic concept is that the neurons need to be activated synchronously at the same time, and they are connected. It is why people used to say they fire together. They fire means they get a action potential. They fire together because they are wired together. So it is why in my lab we focus in both, in the activity of the groups of neurons and in the connection. The connection, as you know, they are known as synapses, okay? So in my lab, it is why the name of my lab is Molecular Codes of Memory. This is the name I choose because this is why we are interested in. And we, as I told you, we focus on the epigenetic mechanisms that happens in the nuclei, basically, and in the nodes of connections, in the synapses. And for studying synapses, we use transgenic mouse models for AD, for Alzheimer's disease, human brain tissue from people that die from AD. And here I just wanted to give you some information for the talk. So once every neuron is activated, it is because it received the message. This neuron will get activated. It received the message from this neuron, and how detect the message? Well, in the synapses. So here is the presynaptic area where the neurotransmitter are. The neurotransmitter are released, and the second neuron catch all these neurotransmitters using receptors. The receptors are proteins that sense when the levels of neurotransmitter increase. So that means that these neurons were activated, and this connection increase the activity of the second neuron, okay? Okay, well, I will start with the data, and the first part of the talk will focus on the epigenetic control of memory. And first of all, I will remind you why are important proteins. Proteins are very important for the cell functioning because they have different functions. For example, we have thousands of different proteins in a cell, and they have different functions. For example, we have insulin that allows to communicate, the cells to communicate, and when it reaches its receptor, it allows to get inside the glucose. We have enzymes that that work in the DNA replication. We have some proteins that work as transport, or for example, hemoglobin is important for transport oxygen, and we have also structural proteins. Now you think in the importance of proteins, you will think how the cells control the production of proteins, that they make everything in the cells. So you will remember this class, maybe if you already have the class of biochemistry, you know that for making proteins, the first step is reading the DNA. So there is a gene for each protein. So you have your DNA and you read it, and read it means there will be a molecule of RNA, mRNA, that will transport the information from the DNA to the cytoplasm to codify the synthesis of a new protein. So every protein is synthesized in this way. This is a mechanism of transcription, this part, and translation, this part. In every step, almost in every step in this long process, there is an epigenetic mechanisms regulating this process that regulates the production of proteins. This is why the epigenetic mechanisms are really important. I will describe very briefly. So we have some epigenetic mechanisms that methylate the DNA. So if some regions of DNA are methylated, they are repressed. It can be expressed. It can be for mRNA. We have another mechanism at the level of RNA. So when we have a lot of microRNAs in the genome that are such as this, that binds its mRNA targets. For example, if this will be the target of hemoglobin, if we have a microRNA that binds, that targets this mRNA, the expression of hemoglobin will stop, okay? So, and finally, another mechanism, I will talk in detail about this mechanism, is the histone modification. As you know, the histones are these proteins that coil the DNA. The DNA in the nucleus are not linear. It is packaged. It is packaged in some structures called nucleosomes, and nucleosomes are formed by histones. And if we modify chemically the histones, it allows the chromatin to open or to close, allowing reading or not reading the DNA. So here is a general scheme. Like here we have the chromosome, that it is constituted by nucleosomes, and in the nucleosome, this is the DNA. It is coiled in the nucleosome. These H2A, H3 are the names of histones. Histones are proteins that compact the DNA. If we modify, it is possible to modify the tails of the histones with chemical tags, and some tags induce that the chromatin opens, and some tags indicate are chemical signals for the chromatin to close. Obviously, the mechanism is so complex. It involves multiple proteins, but finally we can simplify this and say like, if this region of the histone is methylated, the signal is for chromatin to be closed. So with these epigenetic marks, we can have different structural and functional states of chromatin. As I tell you, there are some regions with a lot of epigenetic marks indicating or signaling to close the chromatin, and some epigenetic marks that indicate to the cell to open the chromatin. And there are then all the genes present in the open regions can be read, can be translated to proteins, can be reach the final step of protein synthesis. Well, we are now focusing on this epigenetic mark known as H3K9-IMI3, because some interesting findings I will tell you. So in the lab, we are now studying this in a single, but our plan, our long-term plan, is to study multiple epigenetic marks to decipher the code in neurons, how they are controlling the neuronal activity. And we do this also combining we aim to understand how these epigenetic marks control or have influence on learning and memory process, how they are affected by aging in the brain, and finally, how do they contribute to cognitive decline? Particularly, we work with Alzheimer's disease. What is the mark we are focusing now? The name is H3K9ME3, and it means the following. H3 means that this mark happens in histone 3. It happens in the chemical modification happens in lysine 9. Lysine, as you know, is an amino acid. And which is the chemical modification is a 3-methylation, okay? So every time I say H3K9ME3, that means that I am talking about this epigenetic mark. It's a repressive mark. It controls the closing of chromatin, okay? It represses the genetic programs. And another important concept for this talk is this enzyme. This mark, this 3-methylation, is obviously, as everything in the cells, produced by an enzyme. So there is an enzyme in mammals that it is called SUB39H1. I will refer to this enzyme for short as SUB, okay? SUB increases the levels of 3-methylation in this site, okay? So the chromatin pass from no methylation to 3-methylation thanks to this enzyme, okay? So in the lab, we are interested in control this enzyme, but multiple strategical experimental strategies to see what it does for brain functions. So why we are interested in this mark? During my postdoc, like 9 years ago, we discovered that in the hippocampus of mice, when mice get old, the global levels of this epigenetic mark increases. Meaning maybe like the chromatin is closing in this area that it is critical for memory, okay? So 3 years later, an independent group confirmed this finding using another technology known as Western blot. They found that when they compare young, adult, and old mice, they found that the H3K9ME3 mark increases. They also found and reported that SUV also increases with age. So suggesting that this is consistent with our previous finding. Here, it's important to tell you that here we were measuring the H3K9ME3 levels as a whole, because we use a technique called ELISA, in which that means that we homogenize the hippocampus. We dissect the hippocampus from an animal. We homogenize and detect the global levels of the H3K9ME3, regardless of if they come from glia. You know, in the brain, there are more than neurons. Are neurons, microglia, oligodendrocytes, astrocytes, different types of cells. So we found here that the global levels, we don't know in which cells, increases with age. It is why we started here in Mexico from this finding, our research, because we were interested in which cells this epigenetic mark increases, was our first question. Something also important is that in 2020, a group described that the levels of this repressive epigenetic mark increases in the brains of people that died from Alzheimer's disease. You can see here the staining. This is a coronal section of the brain in which each dot represents a cell, and the color, the intensity of color, relates to the level of H3K9ME3. And as you can see, compared with people with diet without cognitive decline, people with Alzheimer's disease has elevated levels of this mark. We can considering our findings and this finding, we can think in a model in which when the H3K9me3 levels increases in the brain, there is an increase in the condensation of chromatin, and very important genes for neuronal function stop to be produced. And finally, this affects the connectivity, the synaptic plasticity. This is a working model we are following now. So with this in mind, we designed this experiment. We injected mice, aged mice, like at 18 months age old mice, and we injected with an inhibitor of this enzyme, SUV. We confirmed that when the animals were injected with this pharmacological inhibitor, the levels of H3K9me3 get down, okay? But surprisingly, when we do that, the animals, when we analyze it in detail, these animals, we found that the levels of BDNF, that it is a neurotrophic factor, is like a food for neurons, increases. Also, we found an increase in the total number of synapses in the hippocampus. And very more important, when we run 3 different types of tests of memory in mice, that one is unsupervised learning, object location memory, and field conditioning. All of these are tests or tasks that we can run in a lab to evaluate memory. When we run that, we found that animals with low levels of H3K9me3 has higher performance, improves the performance of memory. So with this evidence, we decided to investigate in detail all of these findings. So we are using now, this is our main technology from our research, and it is known as flow cytometry. Flow cytometry allows you to analyze each neuron individually. You pass from the device thousands of neurons from any region in the brain, or thousands of synapses, and it will be analyzed each by one by one, at the single event level. So we have a laser to identify which markers they have, because we have fluorescent markers with different colors, and markers, I mean, which proteins are expressing these synapses or these neurons. And for sure, we choose markers that are important for memory, okay? So, and also important in this technology is that you can measure multiple parameters at the same time in each neuron, and you can run 1,000 per second. The analysis is really, really efficient. So using this technology, for example, in the experiments I described previously, I found that this molecule, PSD95, it's a critical molecule for clustering receptors in the synapses. So when we measure the levels of these important proteins in animals treated with vehicle as control, or ETP69, that was the drug that was inhibiting SUV, we found that treated animals has increased levels of this protein, okay? So also we measure another markers and found the same. So it suggesting that this, if we inhibit the enzyme that is closing the chromatin, we can get more functional synapses and more efficient memory. So the summary of the findings up to here is such as this. We first found that the hippocampal neurons accumulate this epigenetic mark during aging. In aged mice, we tested that if we pharmacologically inhibit SUV, we can reduce the levels of H3K9me3 in neurons. We increase the levels of this neurotrophic factor. We increase the number of synapses, and we rescue memory. So the question next was, what is the functional impact of the H3K9me3 accumulation that happens in aging in the neuron activity? Was our question. And we decided to run the following experiment. Our hypothesis is as follows. H3K9me3 accumulation reduces neuron activation. This is the hypothesis we want to test, and I will show you some experiments we have. why we support this, how we support this hypothesis. We think that when the chromatin is closed, there is a repression in gene expression, there is lower levels of new synthesized proteins, and it impacts the neuron activation, okay? So for doing that, we run this model. We have, use the novel environment paradigm. So in this paradigm, as you know, the lab mice always live in a really boring cages. So this is a home cage. When we put an animal for 15 minutes in like a Disneyland context with toys, tunnels, and different objects and some walls for climbing, for 15 minutes, it induces a neuron activation. So, and thereafter, we put back the animal, and 1 hour later, we dissect the hippocampus of these animals to see what happens in their brains. Here, it's only an example of how the arena looks. So here are the animals exploring this arena, and we confirm that we track the activated neurons with a protein that is known as ARC. This is an activity marker, so that means that each of these dots are neurons that were activated by this stimulation. So first, we confirmed that our model works. So we compare the animals in the novel environment or animals in the home cage as controls, and we found that the percent of ARC positive neurons increases by the stimulation. Here are where this data comes from. So we stain, this is a region in the hippocampus. Neon N is a neuronal marker, so all of these in green are neurons. We track at the same time the levels of the epigenetic mark, but this is like, maybe you can see, these small red dots are neurons that were activated when the animals were exposed to Disneyland. And next, we needed to tool for inhibit or reduces the levels of H3 kinase 3, specifically in the hippocampus, not systemically. It's specifically in the hippocampus and with a more specific tool. So we use technology known as RNA interference, that it is like you can inject an RNAi molecule that will target your mRNA, in this case is SUV, and will degrade it. So in this manner, with this technology, we injected in animals these viral constructs. In a virus, you put all this genetic information for producing this RNA that will target your mRNA that you want to reduce the expression. And we do this specifically in a region of the brain that we are interested in. It's the hippocampus. So to do that, you need to do a stereotactic surgery. And that means that you will inject in a specific coordinate. Here is, for example, here you can see the brain of a mouse. The sagittal view is like a, well, and if you do a cut here, you can see this map of the different regions of this of the brain of this animal. And what I am showing here is where we injected the virus. Okay, when we injected the virus, the neurons get green. We can see the neurons that get infected because the construct, the virus, has a how do you say, a reporter, a gene reporter. So every time the neuron produces a green protein, that is EGFP, that means that the neuron is producing this technology to reduce the expression of SUV. Okay. And then we bilaterally inject this viral construct with the idea of reducing the epigenetic mark, with the idea of opening the chromatin and rescue all these normal functions that a young animal have. And in this animal, we put it in the novel environment. And what we found, well, next we use flow cytometry to dissect the hippocampus, to identify the cells, confirm that they are cells with a cell marker, and finally analyzed by splitting the population in those of them that are neurons and those of them that are not neurons. That can be oligodendrocytes, astrocytes. And finally, in these neurons, we can specifically analyze those of them that get green, okay? That express the construct that we use for inhibiting SUV. And finally, in these neurons, we analyze how much of them get activated. And what is the summary of the expected outcomes? We are still working on this project. So we will quantify the levels of the epigenetic marks in parallel to the quantify how many activated neurons get the animal in response to the novel environment. And finally, we will evaluate memory in these animals in which we are controlling this epigenetic mark. This is almost the end of my talk because I will talk about our new method we recently developed, because we have the question, we want answering some question at the level of synapses. So we want to know what happens with the neuron, but also with its connection. And for that, we developed this method called nucleosynapse tag. And why it is, the word tag comes from the idea that we are labeling with the green color the neurons, but also the synapses that comes from that neuron. And we dissect after viral injection, we dissect the hippocampus, we homogenate the tissue with a lot of steps, and finally, we get nuclei and synapse from the same animal, and we can focus on some neurons or nuclei of neuron that was transfected for this construct. And also at the same time, we can track by flow cytometry the synapses that were infected by this construct, okay? And here we have a lot of experimental evidence supporting that we are having synapse and nuclei separately from the same animal. And this is, I think, my last slide. The idea we have now is we have a powerful tool to analyze in parallel the neurons and the synapse that were where we are modulating the epigenetic landscapes, because we will focus on EGFP synapse and EGFP neurons. And in the neurons, we will measure with it's very quantitative, the method. We will measure the levels of H3K9 and ME3, and in the synapse, we will measure a lot of synaptic markers. Okay, and so the summary of the advantage of the method is that it has single event resolution. It is highly quantitative. It allows multiplex analysis. It's a high throughput analysis method. For us, it's very important because will allow us to detect nuclear, in this case, epigenetic mechanisms, controlling synapse structure and function. And well, this is my group, and I just want to thank all the funding agencies that we have for doing our research and our collaborators at UCI. Thank you. I guess it's time for questions.
Yes, definitely. Yes. Thank you, Professor, for this amazing talk. Do you have any questions over here? Okay, so first.
Yeah, sure. Okay. You.
What I
Thank
want
you.
to ask you is that, do you have any type of, or do you do any type of physical proof to the mice? Like, do you make them go through some type of?
If I have somewhat physical or what?
Yeah, like physical proof, like not so much focus in the analysis of the brain, but do you make them do something to prove like the memory?
Like if I exercise the animals?
Yes.
No, well, this is a good question that allows me to tell you that my advisor during my postdoc, Carl Cotman, is an expert in exercise, and he runs some experiments in his lab. after we will discover all of these things. And he have found that the exercise decrease the levels of this mark in the brain by exercising the animal. You know, these wheels in which the animal run. So he is a really expert in that, but he has done that experiment. The answer is yes. Yeah. The microphone, the mic.
Why did you choose to work with mice?
Because the other option is with humans, and I have to dissect the hippocampus. And if there is volunteers, I am so happy. No, I'm kidding. This is a really useful model because it is the kind of answer I will give you is the answer we used to write for a committee of bioethics. So it is it's a model that it is cheap. It is highly consistent because they come from the same mothers and the same parents. And I think the most important answer for this question for me is that we know a lot about how the brain of rodents work. But it is, yeah, I know that it should be better to have a more theoretical models and such as. It is why I choose. It's the best. It's a widely studied model in neuroscience.
More questions?
Thank you. Well, you mentioned that the H3M
K9
K9
ME3
MI3 are the type of histone modification, right? So how are you able to, in first instance, like going to the basics, identify these modifications? Like what do you obtain them from, like a gene bank or data bank or something? Or
Yeah, sure. They have previously been described. People in the epigenetic field have previously discovered that there are a lot of epigenetic marks. I think we don't know all that they are, but this is a widely known epigenetic mark. But yeah, going back, yeah, previously people discovered this epigenetic mark.
And well, you probably mentioned it, but
And sorry, just to be more, this epigenetic mark is present in all the cells in the body. It's all the cells. Actually, it's very important because maybe you didn't realize that, and but compacting the DNA is very important because it is why the cells from the hepatocytes from the liver are very different from the cells to the heart. Why they are different? Because they produce different proteins, because they have different genetic programs. How do they are controlled? How the liver downregulates all of these proteins that are really important in the brain, but not for the liver, by epigenetic reparation. So I will make sure this epigenetic reparation gives the identity for the cells. It's important to give their own identity. Okay. So next, your question. Sorry.
I think you answered my question, but I was gonna ask, like, what made you choose that histone modification? But you already answered that it's present in all the cells. But
yeah, it's present. It's really important. It's really important to close all the regions that you want to use. But what it is interesting in the findings I tell you is that when the neurons are aging, it dysregulates this process. So it's important to close some regions, but not everything. So we have discovered that in neurons from aged animals, some very important proteins from the neuron are not produced because with age, the chromatin is closing. So this is like a pathological function of H3K9ME3. But I insist, H3K9ME3 and the closed region of the genome are extremely important for all the cell functions, okay? Thank you
very
for
much.
your question.
I was just going to ask, like, if
little-
your like technique applies only to Alzheimer's disease or to like the other kinds of dementia. I think there's like
The technique of the mark.
y- the like your research.
Okay. No. This is a good one. Thank you. No. Epigenetic mechanisms are it's increasingly a field, a hot field in neuroscience, because we are discovering that even if the neurons are postmitotic cells, postmitotic means that they do not divide again. They suffer from epigenetic change. So there is a lot of data from Parkinson's disease, from Alzheimer's disease, from sclerosis. Yeah, it's an open field. And actually, it is why when we start to study this epigenetic mark, was very few information, but 4 years later, there was this report in which they found that in 80 brains, that this also happens. Because at the beginning, all the data I show you is only in regular mice, no AD mice, because there are some transgenic AD mice. These are wild-type mice, and they, with age, increases the levels of this. But it seems like if you have AD, this pathological increase is even more stronger. Okay? Now, yeah, did I answer your question? Okay. Yeah, sure.
So with this question, I don't know if you can tell us what happens with if, for example, with the ADHD that also affects memory in some cases.
But again, can you rephrase?
With this question that was made, that also my question is that what happens with somebody that has ADHD? How do, because we know that it affect memory also, but
H what?
ADHD.
Okay. Yeah. And what happens with what?
But we know that it affects memory.
Uh-huh.
So does this also with aging different?
It's a good question, but to be honest, I don't know. In these fields, in this pathological trait or disease you are talking about, no, I don't have information now. But sure, it's an interesting question. But yeah, I don't have an answer. Yeah, memory is affected in different. Yeah, actually, in Parkinson's disease, there is also memory problems. Yeah. Yeah, but yeah. But this is good that you think why open your idea. No, that's good. Yeah, I don't have the answer, but it's a good question.
Do you have any questions?
Yes.
One more question is like, what is your goal or like the impact that you want to make in like with your research in the disease, maybe like future treatment or like what will be like your
Yeah.
goal?
Thank you for the question, because one main component I didn't tell you, but one main component in my lab is we want to have a translational approach. Maybe you don't know this translational science. Maybe you can identify, because it's a long-standing definition. There is basic science in mice, in labs, and clinical science in humans for clinical trials to discovering drugs. there is something called translational science that makes a bridge between these two sciences. And what you do, this is what we are doing, is testing if these changes that happen in mice happen also in the brain of people. And it is why we have the advantage, because our great collaborators at UCI, they provide to us brain samples from people that die from Alzheimer's disease or controls. And we are investigating if these changes at the molecular level also can be seen in that tissue, with the idea of identify targets for possible treatments. Because this increase in H3K93 happens in mice, but not in humans. It's interesting, it affects memory, but it is not translatable, cannot be useful for treatments. But if these changes also happen in the brain, that means that probably also are affecting memory. And if we have pharmacological tools, that mean drugs, and drugs, I mean pharmaceutical drugs. If we have drugs that target this enzyme, for example, could improve memory. Actually, I have to tell you that Carl Kottman patented this compound that we are using because it has the potential to be useful in clinic. In clinic means in people for improving memory. Because finally, what we are pretty sure is that if we use this drug, ETP69, in mice, they improves their memory significantly. It's amazing, but it doesn't mean that it works in humans. But yeah, this is our long-term goal, to contribute to something such as this. Thank you.
So,
Thank you for your talk. It was
Mhm.
amazing.
Thank you. You're welcome.
I wanted to know, I don't know if you mentioned, but at what stage of the disease the mice that you are using, like how developed is the disease in the mice that you are using?
How I develop Alzheimer's disease, for example?
Like what's the stage in which they are? Like how strong is the Alzheimer that they have?
A good question, because all the animals I am using, this I will go back quickly, because I was a little bit confused. This animal I represent here, such as these are wild type. But it is known that as in humans, with the animal's age, I mean, when you compare a 4-month-old mice that it is young and it is equivalent at 20 years, such as you, we compare with the 18-month-old mice that it is like a 60 years old in human years. The animal with the younger animal performs better in the memory test. So this is we are used as a model of aging. But we have also in the lab some transgenic mice that we use as a model of AD, and what this animal have maybe you know that in AD there are some mutations in some specific proteins that accumulate in the brain. So these mice has these mutations, has the human protein with this mutation, and the scientists, we have found that these proteins accumulate as in humans, in the animal, at eating these memory deficits very early, like at 6 months old. It's a very adult mice with memory problems. These are the models we use for AD. But in all the experiments I described today, these are wild type, regular animals. What induces the memory deficit was the aging process, as in humans. I think this is not surprising from everyone. You can see that maybe you cannot know, but maybe your teachers have noticed, myself, I have noticed, that I don't have the same abilities for learning as I was younger. You are not worried about that now, but you will be worried later. So, and if you see your granddads or grandmoms, they learn a little bit slowly. And if you compare with some very young, the younger people will perform better in almost all the tests. But it is normal, okay? There, just to tell you, there is a normal decline in function with aging, but if you can draw the decline in aging, in normal aging is such as this, and when people has Alzheimer's disease is such as this, okay? This is different, it's pathological decline. Thank you for your question.
Thank you.
Here, there is another question.
I'm sorry, and do you know that if the implementation of SUV to the mice is after the next generations?
If the what?
If the implementation of SUV and
That word, if the what?
the what?
The first word, if the
implementation, like
implementation.
well, when you like put the SUV on the mouse.
Ah, okay, affected next generation. Good question, because I didn't, good question because I can explain better. Sorry, that's the other side. I didn't express SUV. What I express in these animals, this an RNAi for SUV. RNAi means I design it because I can, you can do it with this technology. A molecule such as this that targets the mRNA from SUV. Because it targets SUV mRNA, it gets degraded. So I downregulated the levels of SUV. But your question is good. If this affects the next generation, I don't think so, because the change we are doing is in the brain cells. And when you, maybe you know all of this, that when you, what you need for generate a new human or a new mice? An sperm and an ovule, right? I didn't modify the germline cells, the sexual cells. I didn't modify. I modified.
All for coming. If you have any more questions, you can always talk to Dr. Alep or send him an email. Thank you all for coming, and stay put for all the other events that we will be having this day and in the rest of the week. Let's give another round of applause to Dr. Alep. And we have a small part of our
Okay, thank you. Thank you very much.
Okay, thank you everyone. The next talk is going to be happening at 3:30 today. So we'll see you at 3:30 at A150. Otra vez, que no me acuerdo el título. Debería de abrir, abre hoy o abre mañana.
Déjate confirmar. Building Gallery es mañana.
Abre mañana y el viernes.
Ah,
Sí, es mañana a las 12.
¿necesitan algo para la conferencia?
Ah, sí, está en Ilias José Antonio Cervantes.